Okay, evolution part two. So looking at molecular data, we can use that to draw evolutionary relationships, whether that is from DNA or if that is from proteins. So if we look at our first chart here, each of these letters represents a single amino acid. And if we look at the yellow, that represents everything that is conserved or saved from in all of the species. And the white parts are the things that are different. And so if you look at all of the letters, we can see that bacteria down here is actually the one that is most different. So this would be the out group. And then we see that between human, tomato, and yeast, tomato and yeast have one difference here, and then humans and tomatoes have one difference here. So that would make drawing this cladogram a little bit challenging, because how do you differentiate between those? But that's just part of it. On the AP test, it'll be a lot clearer. It won't give you such ambiguity, but that's how you would look at this to help start developing a cladogram, is looking at those differences. If you are reading a cladogram such as this one, we know that the ones that are farthest out, again, this is our out group. The one with the least difference or the most differences. And as we progress, these descended from a common ancestor here. That is the closest. So these two would be the most closely related. The data for this is coming from looking at the hemoglobin protein in red blood cells, comparing it to human hemoglobin. And so we see that the rhesus monkey, another primate, has the least differences. So that's why they are closer together on the cladogram. The lamprey, a very primitive fish, has the most differences from the human, so it is farther away. Looking at this cladogram, trying to take this data and transfer it down into here. Right here, we're going to have two pairs of organisms that are closely related. We're going to have one out group. So when we look up here, we are looking for two organisms that are closely related and another set that are closely related. And so we're looking at the differences in amino acids from cytochrome C, remembering that is in the electron transport chain. I think I mentioned that once, so of course you remember it. So we see that E. africanus and E. ferus have one difference. So they are going to be one of these two. And we see here that A. forasteri and G. gallus, they have three differences. So that's going to be the other one of these. And so we have to use the rest of the data to figure out who goes here and who goes here. That is going to be done by figuring out which organism goes here first. And so this is going to be our out group, the one with the most differences. And so when we look at our data, we see that D. polyeptus is going to have 21 differences from E. ferus, 18 from G. gallus, 20 from A4 from that one, and 20 from E. africanus. And so that's the one that is most different from all the others. So D. polylepis would go there. And so figuring out if this is the E. africanus and E. ferus, if they go here or here, we look at the rest of the data. And what we see is, compared to the D. polylepis, the ferus and the africanus, they have 20 and 21 differences. And we see that D. polylepis, the gallus, and the A. forasteri, I'm going to guess that's hard to read, they only have 18. So that means that these two, which are going to be together, are closer to D. polylepis than these two. So that makes me think that the gallus and the A forsteri are going to go here because they have smaller differences. And the two E ferris and E africanus are going to go there. Now, does it matter that the G goes here and the A goes here? It does not. They can be flipped. Same with those could be flipped as well. But looking at this, the reason that we're putting them on this branch, again, is because they have a closer similarity to them. Okay, so then the highly conserved proteins that we're often looking at, so what it means by highly conserved, this means that these are proteins that are going to be very common to the organisms that you are comparing. So it is important that these particular proteins stay unmutated. So highly conserved means low mutation rate. Because histones, we remember, organize our DNA. Hemoglobin, that's for carrying O2. Cytochrome C is part of the electron transport chain. And then our ribosomal subunits are for protein synthesis. All of those absolutely critical for life to function. Therefore, they are going to need to be highly conserved and not mutated. So that's what makes them excellent things to compare rather than, the proteins that make up hair color. Highly mutated would create lots of complicated data. And so having a little change makes it easier to compare things and how they are related. And it's not just the proteins. This could also be the DNA that codes for those proteins. So often you will look at the DNA, the genes that code for the ribosomal subunits, because DNA evidence, remember, that is actually even more specific than protein evidence, because you can have a identical stream of proteins or identical stream of amino acids, but it's not actually the same DNA because of the wobble effect in the when you're going to that third nucleotide on your codon sequence. All right, human impact on the evolution of other species, why we are so awesome. We kill things, extinction, so whether that is just simply hunting them to death or cutting down their environment, or maybe we introduce a new species which makes them overpopulate and drive out others through competitive exclusion. We use lots of antibiotics and we use lots of pesticides. And so this can lead to both antibiotic and pesticide resistance, gets rid of the weak ones, makes strong ones. Climate change is going to cause the climate and the environment to change too quickly for the species to have the chance to adapt. Artificial selection, picking the traits that we like, that is going to cause rapid mutation, rapid change, rapid evolution. Cows used to be much smaller. Chickens used to be much lighter. Some chickens can't walk because we have bred them so aggressively to make meat that they are too heavy. Domestication does the same thing. We are isolating specific traits, and that is causing not only better traits for us, but then we are removing those organisms from the environment, changing the evolution of those communities. This is a very minor difference that I have never seen show up on the AP exam, but when I was looking over the review guides, and they said differentiation between phylogenetic trees and cladograms. So cladograms are technically more general, and so the length of these branches don't specifically represent time as well as a phylogram. And you're like, what's the difference? So you can see how these lines, just like these two, but they are different lengths. So that means that the evolution of this eukaryote took longer than this one did. But you would still interpret it the same way. So eukaryote 2 and 3 are more closely related than either one are to number 4. 4 is more closely related to those than they are to number 1. All the eukaryotes had a common ancestor back here. And then everybody had a common ancestor back here somewhere. And so that would be right there. The interpretation is still basically going to be the same, but I wouldn't ever think that there's going to be a specific question. The only thing that I could imagine them asking is that looking at these two, which is more accurate? The answer is this one, because this is more general in its schematic. All right, so then using this, looking at these things, what's the outgroup? This is our outgroup, which is the closely related species. Well, that's a good question. This is one of the problems with cladograms. They may show this and you're like, well, that and that's the same size and distance. So which ones are actually more closely related? The answer is these, because if you think about it, these are both mammals. So they're going to have a lot more similarities than birds and reptiles, even though they did have a common ancestor. And so then what's a node or a speciation point? That's where the branching happens. So all of these are nodes. That comes from botany, where branches come off of main branches. In botany, that's also called a node. And then the derived characteristics, those are the things that cause those speciations. All right, so then we get to our biological species concepts. And so these are other species concepts that some scientists have tried to come up with, because our biological species concept is not perfect. But remember that this is a group that have the potential to interbreed in nature. So when we breed lions and tigers in zoos and make ligers, it does not count. And we are making viable offspring. So when donkeys and horses have mules, they're not fertile, they're not viable, that doesn't count either. And so we have to make babies that can make babies that can make babies. The morphological species concept is focusing on kind of like how things look. We already know that that's not super accurate. This is how they play with their roles in the ecology, even a little more vague. And so we're going to focus on the biological species concept. The problems with that, it does not address how we would then separate things that only reproduce asexually, because if the definition is how they breed, then that doesn't help us. Things that look alike, that can breed together, wolves, coyotes, and dogs, separate species, but that doesn't fit the definition. And then you do have times when things that we have as different species breed and make viable fertile offspring, but we still call them separate species. And so again, this is humans trying to put rules on nature, and nature gives us the double finger back. So double middle finger, by the way. So then moving to looking at evolutionary evidence, you have gradualism. So this is just kind of slow, constant change. And then catastrophism and punctuated equilibrium. So basically you have stasis, nothing is going on for a really, really long time. And then something bad happens, kills a whole bunch of things. That's why you get all of the fossils at once. And then you get rapid evolutionary change. And the next time some catastrophe happens, you get a bunch of fossils. And then, but during that time, you can have lots of changes going on that don't necessarily get fossilized. You talked about that already. What does the fossil record sometimes show gradualism for some species? Most species kind of support this idea, but both of those are interpretations. Things that could lead to punctuated equilibrium, that is the catastrophism idea. Again, changes in non-fossilized tissues, behaviors, intelligence, whatever. And then you can have rapid change when we change our homeotic or the Hox genes. So these are the genes that regulate development. And so we have all of these genes. Each of these genes like kind of correspond to different sections of the body and how they develop. So imagine that this gene gets mutated and then your arms are suddenly T-Rex size and you're just like, that'd be a rapid change in fossil records if that happened. And remember, we see this when as we alter Hox genes in mouse embryos, it changes how their paws develop. And the more that we alter, the more they kind of go backwards into that fin-shaped form. And then there's that recent study published that one change in a homeotic gene caused the zebrafish to grow eggs. So that's very rapid change from one mutation. All right, so then our basic types of evolution. So divergent evolution, you have a common ancestor, and then over time you get kind of large scale changes. So this is going to be macroevolution for a long time. Adaptive radiation, this is more microevolution. And so this was your common ancestor that landed on the Galapagos, and then getting into our different areas, it changed. And so this is typically fast, but again, we were talking geological time. So this could still be thousands of years, and that could be hundreds of thousands of years for that difference. So looking at this, again, adaptive radiation, micro, divergent evolution, longer for macro, and just a couple examples like that. Different ways that things will speciate. You can have sympatric and allopatric speciation. So our allopatric, we are separating, so you get a mountain range, grow, plate tectonics, etc. And then the species differentiate. If it's sympatric, it happens in the species. So sympathetic, you feel for someone, same things. So that's within the population. That's usually due to some type of mutation that isolates it for breeding. Different visuals for it. So allopatric, something is separating the species, so we get a new one. And then sympatric, you have reproductive isolation happening within the main population. These are some finely tuned variations of allopatric speciation, this peripatric and the parapatric. I don't think that these will show up because they're basically the same things. So this is literally a physical barrier for allopatric. Peripatric and parapatric, this is more of what is known as that peripheral isolate. So peri for peripheral. And so it's right on the edge. So it's a slightly new niche on the edge of the main population. So then they get genetically diversified and become a different species. Same thing over here, parapatric. Kind of think that is parallel or right next to. Both of those, they're basically the same thing. Realistically, what the AP test is going to ask you is allo versus sim, and it might talk about the peripheral isolate, which is a form of allopatric, kind of midway between these. So I wouldn't get too worried about it. Just know your allo and your sympatric. All right, some species barriers, things that keep things from becoming the same species. So habitat isolation. So if we've got some snakes that live in the grass and snakes that live in the trees, they may not breed because they don't run into each other. Temporal isolation, so something breeds in the fall, another species breeds in the spring. So the ovulation cycles of the females are different, so they're not going to interbreed. Behavioral isolation, so, different breeding songs, different mating behaviors, keep things isolated. So there's different birds of paradise. The female who is super particular about its own species is going to completely ignore another one. Mechanical isolation, the parts don't fit together, so we're not going to make some babies if things can't connect. And then gamete isolation. So again, this is going to go back to that the protein receptors on the surface of the egg do not match the proteins on the surface of the sperm, so they don't fuse. If the sperm and the egg do fuse, we might have some post-zygotic barriers. So sperm plus egg equals zygote. If that zygote dies or is very weak, that is called reduced hybrid vitality. So it might hatch, but then it dies very quickly, or maybe it dies in utero. Reduced hybrid fertility. So again, mules can't reproduce. And then hybrid breakdown is they might be able to reproduce once, but then their offspring are sterile. So ultimately it keeps those species separate. So all of these are post-zygotic because a zygote did form, but then it doesn't continue on to make viable offspring. So there are also some major extinction events. You do not need to know any of these. You just need to know that there have been 5. They're interesting to read about if you want to take the time, but how, the growth of new organisms like plants affected things, how some plate tectonics did, then you've got your meteor event. But we are now currently, according to some ecologies, in the 6th extinction event because of a certain species, not necessarily an individual, but us as a whole. We are wiping out lots of things very, very quickly. Extinction can be good. If you get rid of one species, it can open up a niche for others. So we talked about competitive exclusion. So if you get rid of that strong competitor, then that weak competitor can move in and have a larger niche. However, this does lower the genetic diversity of the community. So if you have fewer species in a community, so going back to Simpson's diversity index, if you have fewer species, you have a lower genetic diversity, and that reduces that environment's genetic resistance to change, because you have less things to occupy, fill niches, and do those ecological roles of producer, consumer, decomposer, etc. Okay, back to our prebiotic evolution. We have, again, the Earth is present, and we think that there was lots of volcanoes. We had lots of UV. We had lots of heat. We had water. We had lots of gases, but we did not have O2, and all of that gave us good environmental conditions for the abiotic synthesis of lots of our chemistry. So this is called prebiotic chemical evolution. So making the amino acids, making the nucleotides for nucleic acids, making those phospholipids for cell membranes, and then they all start kind of clumping together. And of course, that protobiont, that very simple cell, those are going to trap things on the inside, creating that separate internal environment to potentially give it metabolism. And so then you start having these things start to bond together. So again, the role of clay as an inorganic catalyst that can drive dehydration synthesis and the formation of polymers. And then, of course, we get to the RNA world hypothesis, where RNA is our first genome, but also our first enzymes, because we know ribosomes can act as enzymes and they are mainly made of RNA. So sometimes called the ribozyme. And so then we get that RNA inside of a protobiont, and then you get LUCA, the last universal common ancestor, which then over time starts to diversify into life. At some point, DNA becomes popular because it's more stable than RNA. This diagram, your prebiotic evolutionary environment, modern environment, lots of O2 because of the evolution and development of photosynthesis. And then there is the panspermia idea that maybe everything came from somewhere else, and then it's like a big giant life pod, and then it just started evolving differently on Earth. That just pushes the idea of where life came from somewhere else, doesn't solve the issue. This is our Miller-Urey experiment. So what they showed is abiotic synthesis of your amino acids, your nucleic acids, and ATP. So they showed that all of the stuff we need to build life, we don't need life to build it, but then it can come about. So then our RNA world hypothesis again, and also RNA can self-replicate, so it can make copies of itself, and so then that's basically heredity and basically life. All right, there you go. Nice view of my head as I stop.